DNA wants to replicate. So in order to do this, topoisomerase comes in and unwinds DNA from its double helix form. And then once it's unwound, helicase comes in and breaks apart the two strands of DNA, while single-stranded binding proteins keep the DNA strands from unwinding back up.
RNA comes along and brings in the first RNA nucleotide on the leading strand towards the replication fork. Then DNA polymerase brings the nucleotides in.
Notice how C always binds with G and T always binds with A. On the lagging strand, RNA primase brings in RNA primers that start Okazaki fragments, and then DNA polymerase 3 brings in the continuous nucleotides that build away from the replication fork. And it continues in this pattern on down the strand. And notice that I left a space in between the two Okazaki fragments because of what will happen next.
So, ligase comes in and brings in this nucleotide to fill this gap. And then DNA polymerase 1 replaces the RNA primer with its corresponding nucleotide. And again, ligase fills this gap.
And on this lagging strand, we have a new strand of DNA.
Then DNA polymerase 1 comes in and replaces the RNA with DNA.
And we have two new strands of DNA.